Good afternoon, everybody. Welcome on our monthly Gateway to Translation webinar series. My name is Rievaj and I work as a senior research facilitator in Cambridge Academy of Therapeutic Sciences. Cambridge Academy of Therapeutic Sciences is an interdisciplinary research center of the University of Cambridge, established to facilitate the development of the new therapeutics and support the education and training of the next generation of the researchers. So we are pretty active in translational research, which means the development of the new drugs, new therapeutics, new diagnostics, and new medical devices. And we have a few activities to support the researchers at our university, including direct funding, access to the expertise needed for the development of the projects, as well as educational resources. So one of the things which we are doing is this monthly Gateway to Translation webinar, where we are inviting people from outside of the academia to talk about the new products or the developments or abilities of the industry or the new way or the way of thinking in an industry, which is often quite different from the academia. So if you want to know more about our activities, on the right side, there is our website, ATS CAMES UK. Please feel free to have a look. We run this together with the Translation Manchester, which is the part of the University of Manchester, and their main mission is similar to ours, to support the translational research in the University of Manchester. Again, you have the website there. Today it's my pleasure to introduce a current speaker, Dr. Alan Griffith. Dr. Griffith obtained his PhD in biotechnology, and that followed with more than a decade of experiences in various roles in the cell and gene therapy space. Over this time, Dr. Griffith managed to raise more than 50 million in VC funding and angel investment, and was granted various patents in the AAV design and route of administration approaches. He has been pivotal in enabling more than 3 clinical trials across 5 different disease areas. And when working with Meira GTX, which was a spin-out from the UCL, Dr. Griffith went on help to design and build London's first end-to-end multi-vector, multi-product GMP facility. Currently, Dr. Griffith works as the head of the global operations at VectorBuilder Limited. VectorBuilder is a global CDMO company providing complex support for the development of the gene therapies with an integrated platform to assist researchers and biopharma to mitigate risks when developing gene delivery drug pipelines. In addition to his role, Dr. Griffith is also an advisor to many industry groups and has an experience in delivering lectures for both UCL and King's College. So, Dr. Griffith, the space is yours.
Thank you very much, Juraj. Very good. Nice to meet you all today. I'll caveat this whole talk, which the topic is Don't Play Roulette with Vector Candidates, Rethinking Vector Design and Delivery Methods from the Lab bench to gene therapy in a clinical setting. Today, I'm obviously representing VectorBuilder, but this is not a sales pitch. This is very much about me and my time as a drug developer, where ironically, there's not many people go from being a drug developer into a contract development manufacturing organization, or a CDMO for short. So what I try and do is, we deal with thousands of clients. What I try and do now is bring the 15 years of drug development experience to the CDMO, which means that I facilitate thousands of our clients. This is just a portion of that where we are really looking at vector design and optimization around the manufacturing for these vectors to increase your chances of success. So I'll wanted to caveat that, because obviously representing VectorBuilder, however, I am giving my understanding from the point of view as a drug developer. This is just a brief disclaimer. Again, I don't expect everybody to read that. I will go through, this is the agenda points. Again, just kind of keep it as simple as possible, but I think I would like to tee it up nicely with some gene delivery background, a history of cell and gene therapy, a slide on each, and then some headwinds and tailwinds. These headwinds and tailwinds are very important for the purpose of this talk, because some of these headwinds are the reason that we have to rethink vector design. For those of you who may or may not know what a vector is, I will briefly discuss what a vector is, just in case there's people in our audience that do not know. I'll go through the problem statement, again, why it is imperative to rethink vector design and why it's important, not just from a performance and efficacy perspective, but manufacturing and safety as well. I'll explain where CMC and GMP processing fits in. Again, these are terms that you may or may not know. CMC is basically just a short acronym for chemistry manufacturing controls, which means manufacturing. So again, this is about education. It's about letting people in the audience know that we really have to think about manufacturing on these vectors, not just from a research setting, but from a clinical scale-up manufacturing position. Fourth agenda point, I'll talk on some example case studies and use some data examples just to show you that it's more than just a verbal concept. It's actually put into practice now, and it's actually utilized by some clients and ourselves internally. And then I'll finish with some concluding remarks and just a repeat and circle back to some of the points I've made, the important points that I think are important to share. And then we'll finish, as Juraj said, with a Q&A session at the end, okay? Very briefly, unfortunately, it's very hard to put together a presentation like this without using acronyms and abbreviations. Unfortunately, I'll try and use them minimally where possible, but these are the main ones that I think hopefully some of you will know. I'll quickly run through them, but GMP is good manufacturing practice. These are well-known terms. These are not my own terms. TPP, which is target product profile, which is ultimately what you need when you're bringing a product to the clinic. You need to have a target product profile as per regulatory requirement. ATMPs are just an umbrella term for cell and gene therapies. Again, it's advanced therapy medicinal products. Again, just an umbrella term. CMC, as I touched on there, this is probably the one I wanted to highlight the most because it is to do with the manufacturing and the manufacturing at scale. Basically, if you leave one thing from CMC or learn one thing from CMC, you basically don't have a clinical product if you cannot make it reproducibly and robustly. So this is not me exaggerating. This is a real life situation where you have a product that works really well, a vector that works really well, but if we have problems manufacturing it, then you never will be able to realize it in later stage clinical trials or ultimately a commercial product, which means that you cannot get money for it. You have to expend a lot of money on manufacturing costs, which are prohibitive. All of these things make the whole sector, it's a really big issue within the sector, hence why I've highlighted it. I mentioned cost of goods there, COGS. This is the cost of manufacturing, as everyone knows. You keep that as low as possible in order to make the product as accessible as possible. AAV is a vector. It's one of the many vectors that we manufacture. I've used AAV as an example vector form throughout this presentation just for ease, rather than swapping and chopping and changing. Adeno-associated virus is the most common vector used in clinical trials right now. It's about 79-80% of total clinical trials are taking part with AAVs right now. That is kind of changing for various reasons, potentially due to the size of the genome, but I won't get into that today. That's not part of the scope. CTA, clinical trial authorization, that's a rubber stamp from the regulatory authority that you need in order to conduct a clinical trial. And then IND, an investigational drug, which is the first part of an application process when you're creating proof of concept data, which potentially many of the audience will need to do in the future. So again, I've ran through it quite quickly, but I think it's important to kind of touch on them because I mentioned them quite a lot in the presentation. Okay, very quickly, again, I think a lot of people in this room will understand this. Gene delivery, basically putting any genetic material inside a vehicle. Again, more often than not, we use viral vectors. The whole world is changing a little bit now. We're seeing the advent of more non-viral gene therapy vehicles coming more and more. However, they are just a vehicle or a conduit to achieve the same thing. On the right-hand side, it applies to life sciences, genetic medicine, agriculture, green energy, industrial fermentation. The same applications apply. It's basically just putting a foreign piece of DNA somewhere where it's not usually. Quickly going into the history of cell and gene therapy. Again, it obviously has grown quite naturally with the advent of the DNA double helix for Watson and Crick, but obviously then going into gene therapy being the first option due to recombinant DNA, where you can actually cut and paste bits of DNA into different vectors. That's pretty much, you wouldn't have gene therapy if that initial discovery didn't happen. So cell and gene therapy now has had an exponential growth potentially, you could argue, up until basically 1999, where the first few clinical trials that were using lentivirus, for example, we had the death of Jesse Gelsinger, which is the first patient on record to die from a cell and gene therapy product. That really stopped the whole field in its tracks for maybe about a decade. People were still doing research, but clinical trials had pretty much stopped. There was no investment money, there was no real progress, and it pretty much was just in limbo. Fast forward up to 2008, 2009, people start realizing that you could create better vectors, safer vectors, spending time designing vectors more. You don't have to use the very first design that you use. You should really think about it. Why have I told you that today? Is because you've gone through this wave, peak and trough of gene therapy already. We are already, after experiencing a down trough, we're coming back up to the top, and we're now in the middle of a bottom trough right now, honestly, because there's a lot of clinical trial holds on right now. There's a lot of poor performing clinical trials. We have a perfect time now to introduce again the idea or the concept of better vector design. It's pretty much limitless, so therefore you can have waves of vector design. I will mention COVID obviously caused a bit of a blip in the entire therapy field because you had a lot of issues on supply chain, logistics, but ironically, because of the COVID vaccines, gene therapy actually got a bit of an influx of money then post 2022 because you had pretty much validation that you could generate ATMP, or you get viral backbones or adenovirus in the case of the Pfizer vaccine, that it actually worked and you can actually deliver it to large portions of the population. So due to COVID in a weird way, it really did help cell and gene therapy sector overall, even though it did cause a blip in the industry. It's not all great. There's been obviously a lot of good clinical programs that have gone on to be commercial, of which there's about 28 commercial. That might seem like it depends on where you lie, but that's not a lot. If you think of small molecules and monoclonal antibodies, they have thousands. Cell and gene therapy, very few commercial products in relation or relative speaking about the other modalities, but it is increasing. And to be honest, in the last 20 years, it's gone from 0 to 28. So there is this huge growth. But it's not all good because there has been some withdrawn from the market. Again, this is reason for my pitch today, is why were they pulled from market? They were cited as not being commercially viable due to the cost of goods being extortionately high, the manufacturing cost too high, it was too hard to get the patients in. So therefore, at a certain dose, the manufacturing was too high. Therefore, they got withdrawn. What does that mean? That means the only solution, you have to go back to the very start with the vector design and optimization to really decrease the dose in order to decrease the manufacturing cost of goods. That's the only play we have. That's the main message of this whole project, why you have to reduce the cost of goods for manufacturing in order to make it more accessible commercially, because these products are like £1 million per shot. Only rich countries can afford them. That's a big problem. We need lower and middle income countries to be able to afford them. So what do? We have to revisit vector design, and hence the reason for the talk today. This is just a small location or geography breakdown of cell and gene therapy. You can see, again, China, it's kind of hard to understand what's going on in China due to various reasons, but there is a few products in China as well, and that's okay. Okay, and now we're just going into what ATMP development, so again, cell and gene therapy development typically looks like. You've got this period of time in preclinical. As you know, you have candidate selection. You have to pick a candidate. You can't bring 20 products into clinical trials because it costs millions of pounds to do phase 1, phase 2, phase 3. So you have to pick one. So you're pretty much putting all your bets onto one vector design ultimately. So hence the reason for Roulette. You're putting all your bets onto one candidate, one construct, one design. we have no choice. That's the way we have to do it, unless you have millions and millions of pounds to bring multiple candidates to multiple phases of clinical trial, which even the richest companies, the biggest companies cannot even afford to do that. One, why would they? It's very inefficient and it's not good use of capital money. So what is the solution? Again, as I said, you have to go back, spend time during your preclinical stage for candidate selection, and I'll explain why this is a bit of an issue. You have investors and you have people who are investing in these companies, or grant agencies investing in these academic or industry companies, and they're telling people, please have a candidate selected as soon as possible, 6 months. So the clock starts then. You've got 6 months from then to design a vector. And as everyone knows, they're all scientists here, you cannot do much validation and preclinical in 6 months of work. But this has been the playbook for the last decade. I've been part of this. I was working under duress from a lot of a lot of board members and a lot of investors, and that's fine. I didn't know any better, but now I know that we have a responsibility as developers to explain to the investors, to explain to the board members that we should extend the candidate selection runway. A lot of people understand the cost or the money runway, the budget runway. Now it's time we talked about the candidate selection runway and extend that in order to save huge amounts of issues, clinical holes, and costs later. So that's really part and parcel. I'm choosing to show how to explain the same message in a few different ways, because I realize as time has gone on, when I'm telling this story, some people resonate with the way I've shown it here. Some people resonated with the financial side. Some people resonated with the preclinical side. So some of these slides are repeating the same message, but in a slightly different way to help people understand exactly what I mean. And again, it all comes down to speed, cost, quality. You can't have all three. This is the perfect triangle, as everyone knows. And then one other caveat I will explain. This is the rough breakdown on the pie chart of all the different development disease areas that are ongoing. As you can see, cancer takes up a huge portion. I will highlight eye disorders because that's where I've come from initially. Eye is a perfect gene therapy target because it's such a small organ. It's compartmentalized. You have a perfect control. If you dose one eye, you've got a perfect control in the contralateral eye. The dose is very low, so the manufacturing costs are very low, meaning entry point and access to ocular gene therapy has been the easiest way to get into cell and gene therapy currently and historically. So that's very important because that testifies or it vindicates what I said earlier, that if every gene and cell therapy had those small dose requirements, some of the issues that we're seeing right now wouldn't be an issue. And ocular gene therapy is a little bit exempt from that because it's a smaller organ and it doesn't need as much dose of AAV, for example. So yeah, we talked a lot about the kind of history there, but what are vectors? Again, they're simply just the vehicles to deliver these foreign genetic materials. You can have different plasmids, different viruses, LNPs, lipid nanoparticles, vesicles, exosomes. These are all tools in our toolkit, all vectors, but predominantly I'm talking about viral vectors today. Pretty much analogous to anything I've said today will be repeatable for any other vehicle or any other vector, and that's useful to know. It starts with a viral production, of course. It starts with a precursor sequence. As you know, you cannot make a virus without putting the plasmids that code for that virus in a bioreactor or in a cell culture, in a flask. You can even make it in a saucepan. If you really wanted to be adventurous, you can make virus in your saucepan in your kitchen, but I wouldn't recommend it. So it's one of those scenarios where you get all the different components. I've highlighted it this way because it shows the complexity. On screen, you have all these sequences, these DNA elements, okay? They all need to come together with a wonderful stoichiometry that would manifest as manufacturing a virus particle. And that virus particle contains a piece of DNA genome or an RNA genome inside of it, which is your therapeutic payload. So the product for viral vectors is a little bit complex. You have the outer shell, the viral shell, but you also have the inside payload as well. That's what makes these products very, very good, but it also makes them very difficult to manufacture because you've got two things to consider. This is just a breakdown. Again, I am a manufacturer previously. I am a CMC lead. I don't want to get into the nuances of it. Again, this is purely just to tell the story that it's very, very complex. You have your triple transfection normally, which is the most common form of manufacturing. You put it into HEC293 cells. Typically, you have other cell types. Again, then that simply makes your viral vector. So it's simple, in simple terms, it's in the purple box here. But then you, in reality, at scale, you have these upstream and downstream unit operations. So USP and DSP, basically you break the process up into upstream where the virus is harvested, and then downstream is all your purification and all your polishing. For research grade viruses, you don't need to worry about too much of the downstream because these are very expensive equipment. These are very expensive tools to do purification. And for small scale lab work, you don't need the virus to be overly pure. You don't need it to meet certain specifications like you do at GMP or clinical batch stage. And that's very, very important as well, because you have this huge gap in knowledge or in viral profile, if you will. The manufacturing profile is very different. You have a research profile and then a large scale GMP clinical batch profile, and they're manufactured in very different ways. And then, as we know, the manufacturing process means in the final vial, you have different profiles of your virus, which means different empty and full packaging, different impurities, different levels of sterility, different levels of impurities. So this is really the main reason for explaining it this way to you. It all comes down to vectorology. What are the vectors? Why do we need to optimize them? Why is there so much optimization needed? Basically, this is the take home. If it can be modified inside of a vector, it can cause an effect to large scale manufacturing, or it can cause an effect to your clinical outcomes. Okay, this is it. You have to really create a novel DNA arrangement. Again, you've all these different elements that you can change. I've just shown an AAV genome here, ITR to ITR, which is your payload. You can have introns, you can have enhancers, you can have promoters, different Kozak sequences, different transgenes, of course, which are targeting different diseases. You can have downstream transcriptional elements like a WPRE, and you can have many different types of poly A. You could argue that the poly A shouldn't make much of a difference, but I've done large scale studies to show that if you change the poly A even by a few bases, you get huge manufacturing differences in yield and in potency of your viral vector. So even by changing something you think is insignificant causes huge issues in your viral profile. To be honest, let's think about it. How do you choose this? You've got one vector, back to my example of picking one candidate. You have all these variable metrics or elements to choose from. How do you go about choosing that? That's the question. I pose it to all of you. Where would you start? Of course, you start with the basics, but then how do you go through the iterations to create the payload that's going to manufacture really well, be very safe, low dose, and have high performance or efficacy and potency? That's the problem for everybody. And historically, myself included, we used to just make one or two and then hope that it worked in an animal model. Normally it did, but then you'd end up going straight ahead into clinical development. That's okay in some cases, you might get lucky. But what we're trying to tell you here today is we don't want you to get lucky. We want you to go through a quality by design approach where you're designing your vector based on quality from design of experiments. VectorBuilder, for example, we've manufactured 1 million vectors before. Is there a way to leverage that information of sequencing related to manufacturing, related to safety? AI tools now are becoming more and more prevalent. So we're using AI in order to tell us which design is going to give us a better performing vector. All of this is what's happening now, but the field is still a little bit immature. So that's why it's important for me to tell this message today. This is what you need to think about. Again, I kind of touched on a lot of the problem statement already, because that's the why. There's a lot of different clinical holes ongoing right now. It's not just my opinion, it is actually what's happening in the field. You have ATMP failures are attributed to two main bottlenecks. This is now known by the FDA, it's now known by the MHRA, it's known by the sector key opinion leaders. Design of vector is one, and manufacture is the other. Actually, see if I can just use my arrow options. Actually, let's see if I can do this visible. So hopefully that works. So yeah, one can be do with the design, and if you have a poor design, which means that you've gone through proof of concept candidates. If you only have a small budget for a few different animals, how are you going to stress test your different candidates? So basically, it's pretty obvious that the vector design is limited by the amount of designs that you can create and the amount of animals that you have. So this is not people making a mistake where they're being silly or they're doing bad science. This is just a fundamental restriction within the development path itself. So how do we unlock this? Secondly, manufacture, again, as you can see, design and manufacture. But I'll be honest, they're very intrinsically linked. Okay, so it's one of those that they might as well be two sides of the same coin. They are all, because the manufacturing stems from the design now as well. I think years ago, people figured that if you have a vector, it doesn't really matter what the design is or the sequence, the bottom fundamental sequence is. You'll be able to make it. It's just about buying better bioreactors, buying better reagents, better transfection reagents, better media. But now it's been shown that all of those changes, all those improvements, they do help. But the biggest single driver of help is the design. So therefore, that's why it's number one. Again, failures in the news. I was going to populate this with so many, but there was nearly too many to put in here. You've got Sarepta, you've got J&J, you've got Capsidia, you've got you've got Virovec. You've got loads of different failures within the news now that exemplify this. Basically, all the news is telling us that this is what it is. The clinical trial is on hold due to vector design. Clinical trial is on hold due to poor manufacturability of the product, therefore telling us the same story anyway. It's just told a little bit differently in the news. So I'm sure some of you have seen the Sarepta news, clinical trial on hold because the dosing requirement is far too low, so they need to make more of it. So this is the kind of thing that is important for us. Okay, and then, yeah, just changing it up a little bit. This is exact, and just another way, 90% of these clinical trials, if you have, fail. So this is data that's come from global data to show that you have this huge valley of death where it seems like it's quite successful in preclinical. You have a lot of products going into clinical trials. Phase one, around 250 clinical trials every year. Then phase two, roughly about the same. But then you get to phase three and this drops off a cliff. And this is known as the valley of death, where a lot of good products that work don't actually have validity in going any further. So you might have spent millions of pounds getting to that point, and you might have a lack of clinical efficacy, you might have unmanageable toxicity or poor drug-like properties. And then, of course, the lack of commercial need as well, which is probably a key driver for the reason for cost of goods reduction. So yeah, this is, again, really, really important. And again, another way to show this would be the finance, the way the typical flow of products through development normally goes through the valley of debt, which succumb as well to public and private investment money. Because what happens then is investor sentiment is reduced when you have clinical failures, meaning that you might be one or two developers that are doing the right thing, and you can't get investment because people before you have made certain mistakes. And then because you're in the same sector with a lot of the same clinical development steps or phases, you might also have the same issues of not being able to get money then, which is not really fair because you are actually doing things differently. So again, this is the financial burden that we all have that we need to live with. And there's no real way to get around this other than, I said, again, revising and rethinking vector design with a company or a consultation service that has access to millions of vectors that they've manufactured before. Hence why I'm at VectorBuilder now, because that's of course what we do. This is just a real life example where total capital over the last 5 years has been reduced. This means that there's a reduced sentiment in the field of cell and gene therapy. Yes, it's increasing, as in people are going into clinical trials, but the amount of money coming in has reduced hugely. because of those clinical holes. That's really important to show that there's a clear decrease in investment. Once you have a clear decrease in investment, that tells you that we all need to do something different. That's what CliniVec is all about. Yeah, but taking a product to clinic isn't easy. You have vectors from various different sources. Again, this is very important for you guys in the audience, I think, because you're coming from like Cambridge or other universities where you might get vectors from collaborators or other academic institutes around the world. UPenn, Mass, all of these academic institutes have their own vectors over the years, and they normally share them with other academics. Time has shown that these vectors are poorly characterized and they're poorly sourced and they're poorly from origin. It's nobody, again, making a malicious mistake, but these kind gifts are normally issues themselves. So the general rule is don't accept kind gifts from strangers. Create your own from scratch with an industry partner, someone like us or other vendors. And the whole idea there is you'd be able to control the provenance, the origin of your vector. And that's really important for manufacturing, and it's really important for potency and all of that as well. Again, I'll just move quite quickly. I just realized time is creeping on. So, and again, there's a lot of information here, tells more or less the same story, that we want to have a decent patient population. You are looking at delivery. Delivery is something that we won't discuss as the scope today, because you have a vector. How do you make it is one thing. How do you deliver it? Are you going to go directly into the organ, in the heart, the brain, the eye, or are you going to give it systemically? Are you going to decrease the dose by doing route of administration development? That's a whole other conversation and a whole other project. So that delivery example will be left out for today. And of course, we can touch on it. Okay, so now getting into the actual clinical candidate selection process itself, and why it's so important to understand why you have so many possibilities. You can have pretty much 100 million plus possible vector design optimizations before optimizing the gene of interest. That's a scary number. As I said, you have to pick one. So how are you going to pick all of these that impact things like safety, efficacy, and then ultimately the manufacturability? You've got packaging considerations, tissue specific, considerations around the promoter. Do you want to look at novel IP? So a lot of people pick IP as a key driver for doing this development work. They want to create something that's never been done before. So that way they would actually they'd actually want to create something new. So again, that's really, really important. And then, yeah, sorry, I'm just looking through the next slide. Again, sorry, just glitched on me a little bit there. Okay, yeah, now back to where does CMC and GMP processing fit in. CMC and GMP processing is the manufacturing side of things. This gives you a bit of an idea. What do I mean? You've got this scale of manufacturing on the bottom where you go from the flask, or in some cases, some people manufacture it in a tea flask. Not saying it's bad virus, but I'm just saying it will be different virus by the time you get to a large scale bioreactor. You might say, well, I don't need that large scale bioreactor for now. Well, then that's okay. I agree. But you will need it if you're going to have a commercial product in the future based on the current modeling of viral vectors and the current need of the doses required in the field right now. So if you look at all the clinical trials, of which there's like 1,500 open, they all have a high dose requirement. That means you need to manufacture in 500, 2,000 liter scale bioreactor. As such, you're going to have a few issues, and you're going to have amplification of impurities as you scale up. You're going to get then amplification effects where your vector has maybe the payload is not packaged correctly inside. So then what happens is you have subpopulations of the vector, and then the vector will always make those less critical forms kind of more easily. Again, I'm trying to choose words that you'd understand rather than manufacturing terms, but that scalability is a number one killer for a lot of these products. The process equals the product. So again, I might have an AAV, Juraj might have his own AAV. On paper, they look identical, but if we put them to two different processes, two different bioreactors, two different medias, two different reagents, we are going to see fundamentally different potencies, fundamentally different profiles. And that sounds crazy because you have the same sequence 3 sequences being transfected into cells, you'd imagine surely it's a very linear scalar process. No, the problem is it's not a linear scalar process. It has so many moving parts of it. These are living systems. These are living systems within living systems, so bacterial sequences within viral sequences that cause a whole plethora of issues. And that's why I have a job. That's why I'm in CMC. That's why I'm a consultant for CMC companies that are struggling to manufacture these products. And ironically, I used to think the solution was better bioreactors. But now, 10 years later, I can honestly say under-optimized vectors from bad vector design is the reason for all of these manufacturing issues. And this is the hidden cost of under-optimized vectors. These vectors now, you have a chance to normally invest your budget here. This is the whole idea. We want to extend the candidate runway. We want to invest more money in during the preclinical stage or pre-preclinical stage before you get to GMP, the need. Yes, there is a contradiction here. It means you do have to budget more and spend more money early on. But the classic case of spend money now to save money later, this is really, really obvious for this sector right now, that you can really go deeper into telling the investors, telling the grant bodies, telling your academic groups that it's important that you really spend time choosing the best vector that have the long-term manufacturability in place. Again, look, safety and potency is very important, but normally you know it's quite safe quite quickly because you're in animals, you're in cells, you know if it's toxic, you know if you have a toxic gene. Manufacturing is always the afterthought. I'm simply saying bring that earlier, so therefore you have to spend a little bit more money earlier. And not many people are doing this because it sounds, you're breaking the playbook, you're changing the paradigm. This is why I'm having the presentation today. This is why I need the message to go out there. It's becoming more and more well known. Investors are starting to believe in it. Developers are starting to believe in it. It's important then that I think you early researchers also start to believe it, that you don't grow up or you don't develop your career with the idea that the old playbook is how you do things. It's very early. I hope it gathers more traction, but this is the idea that you bring your entry point for your candidate much later after your proof of concept as well. So your candidate selection, instead of doing your candidate first and then doing your proof of concept, why not just do your proof of concept with a bunch of different candidates? Yes, that'll cost more money, as I said, but that is fundamentally the only way you're going to get a better optimized vector to save you, as I said, that manufacturing burden later. You might only need 150-liter bioreactor if it's really manufacturable, rather than a 2,000-liter. The cost difference between a 2,000-liter and a 50-liter is in the millions. So therefore, okay, I'm spending £100,000 extra now, but I might save like £10 million by the time I get to phase 3. And I'm not exaggerating the numbers here. Yeah, this is, I think, more or less the same thing. You can have all these different types of form. The main message is increase your N, increase the amount of clinical candidates that you are designing. Empty versus full. Again, I touched on a lot of that. You have the payload side and the backbone side. We have a lot of good analytics now to show that a lot of these vectors, actually, when you manufacture them, you have a lot of empty particles. That means they're useless. They don't do anything in a clinical setting. So basically, you're paying to manufacture these particles that don't contain your payload. What a waste of time, what a waste of energy, what a waste of money. So that's the main message here as well, that these analytics that we have now are able to actually understand that 10 years ago, I didn't have access to these technologies, so therefore I had no choice. I just used to choose, I think this works well, and hope for the best. Now we actually have access to all of these techniques and analytical tools. Yeah, so the solution, where does it lie? Again, touched on this. The conventional development is you select a candidate up here and you base it on efficacy and potentially a little bit of safety. Well, what we're saying here now is part of, again, we just put a name on it. Again, it has means different things to different people, but CliniVec is what we've called it. It's basically a seamless integration of these 3 cornerstones all together, where you've got extending your runway of vectors. So as you can see, your little, I've tried to do it like a vector roulette wheel. Like these are roulette wheels that you're putting the vectors as the vector design on it, and they're all subjected to good vector practice. So I think the whole idea here is then that you're encompassing manufacturability during the preclinical stage earlier, which means that it de-risks everything else. So there is no other way. I am presenting it with the idea that if I say it to enough experts or enough key opinion leaders, and they have alternative solutions, then the body has come up with a better solution than this. So again, I think that's why we're quite proud that we've come up with something that can actually work. Now we just need people to actually execute and prosecute it. Okay. Okay, going into the final part of our presentation now, just on the time, just double check. Yeah, we've got a bit of time. So these are case studies and examples. The idea here being, again, don't accept kind gifts from strangers. This is a real life example where I've taken a vector from an academic institute, and what you have is an old vector, old version 1, and you've got this real academic product that is highlighted by the regulatory agency as having a load of issues. It's got a load of extra bits and pieces and sequence in there. It's got mutated ITRs. It's got no open reading frame that matches a certain accession number. You've got an abundance of restriction sites in there. And then a big part, it's got an ampicillin backbone, which, as anybody may or may not know, you're not allowed to bring any ampicillin-derived backbones into a clinical trial. But 90% of vectors in the world use an ampicillin backbone. If you're in research, it's fine. You can, of course, use them, but you're not allowed to bring an ampicillin-derived backbone into a clinical trial. And what did we say before? We said that small, discrete changes to the backbone as well make a different vector profile. So if you've done all your proof of concept, all your research work with ampicillin backbone, for example, and then you make the switch to kanamycin for clinical trial readiness, then you essentially run the risk that you have a different viral profile, and you may need to repeat all that research work again. So again, this is where vector design at the earliest stage is hugely, hugely important. That's a really basic example, but an issue that a lot of people fall into, that most of our clients have ampicillin backbone. The number one thing we say, switch to kanamycin as soon as possible before you generate too much data, because that kanamycin switch could instigate a different vector profile, different empty versus full ratio, different impurity ratio. So this was a classic example where we've brought a new vector, and you should, instead of having to do this, like I did, just create the version 2 or your version 1 from a really good starting point initially. So you incorporate all the good design principles, some of what are here, but good vector practice around removing extra sequence, making sure it's clean, making sure that there's no isoforms, making sure that it's kanamycin, making sure that there's no tags in there, that kind of stuff, okay? Case study 2, backbone optimization for plasmids. Again, this is showing pure manufacturability, where we've taken the backbone out and we've ran it through AI generation tool to find what's the best sequence that would maybe elicit the best manufacturing profile. Of course, that's an in silico exercise. You need to do it, empirically as well in in small scale bioreactors, where you validate what the in silico data is telling you. And that's what simply all we've done here. Again, it's a lot of information on the slide, but the main messaging here is that the specific, the volumetric yield and the specific yield, so basically the manufacturing is all about yield. Those two metrics have increased by changing and optimizing the vector plasmid backbone. So if you can imagine those plasmids I showed you at the start of the presentation, by changing the backbone of those, we were able to increase the yields of those being manufactured, and ultimately making the virus then from that also increased. That's brilliant, but you could argue, is that increase enough to bring the cost of goods down enough to make it affordable for a lot of people? The answer is still no. So yes, we're making steps in the right direction in this case, for a particular client, but we also need to go even deeper. We need to expand the in silico tools. We need to expand the vector design, design space, the design of experiment space, and make sure that we don't miss anything. So that's important to know. This was an oversized lentiviral project. Again, using, I suppose, actually, I mentioned AAV a lot. This is a good example of a lentiviral product where we had this oversized cassette where you have a functional titer of 3E7. basically same thing then again, what we've done is we've applied CliniVec process development and CliniVec design modifications, and we've managed to get the functional titer from a paltry 3.6 10 to the 7 up to 1.5 by 10 to the 8 at harvest. And then this was measured by the amount of positive ratio of modified T cells that were actually able to fluoresce. So we had it was under a GFP. So basically we could see that we went from 16% up to 41% by simply changing and applying some of our CliniVec design modifications, which were not huge. They didn't take long. It's not like it was 2 years of work. It was simply a few weeks of extra work, a little bit more time, a little bit more cost at the early stage, cost a few extra £100 of quid in order to actually make them. And the benefits are very obvious then to see when you do downstream analytics. So yeah, I ran through them a little bit quick, but I think they just simplify, explain what I've mentioned so far. Concluding marks, what's it really like? I know we mentioned the 2 or 3 schematics. Here's preclinical development, phase 1, phase 2, phase 3, commercial. But what's it really like? I try to use an analogy here. I like using golf terms because it makes, for me, a lot of sense. But the path to clinic is very bumpy. Golf is renowned as being probably the hardest game ever created because you've got so many variables. Gene therapy suffers a little bit from that versus monoclonals or small molecules. So it's important we understand that, because yes, it has the ability to revolutionize medicine, but because it's still in its infancy and it's in its immature stage, we're still finding that it's probably the hardest modality to bring all the way to clinic. I've just used some analogies here, different clubs. Obviously, that's like the duals, serotypes, promoters, vector designs. Obviously, that vector design area would fall into the different clubs. But the whole idea here is then that you've got different clubs, you've got different obstacles, you've got different courses, you've got different types of swings and strokes, and then you've got different game types. So you got teams, you got 4 balls, you got even the Ryder Cup there recently, which is a different type of game. All of those can be mirrored, as I said, in a clinical setting. The FDA is trying to harmonize all of this, but again, it's very different. And this is another way of looking at it, the types of organizations that are involved, the different courses, the different obstacles, the different clubs, the different strokes, and the different types of games that we can play. It's very, very analogous to golf, if I'm honest. So then this is something that I'm trying to use. It's not perfect example, but it's something that I use to get people to realize gene and cell therapy is not like a conventional modality. It's very, very nuanced and very, very complex, which makes my job very difficult to manufacture them and to create better vectors. But it's not all bad. It's the fastest growing market in biotechnology still, even with the downturn, as I said, some of the troughs. We predict that vector cloning and packaging is going to go maybe from 10 billion to 15 billion in the next 5-10 years. Same for gene delivery CROs, and the same for genetic medicine for CDMOs like VectorBuilder as well. So yeah, the whole projected market has gone up to around 35 billion a year from where it is currently at 15. So for you to say that there'll be plenty of money in the field to iron out these problems and to get over these obstacles is very, very obvious. But the investors still don't want to waste this money. So we still need to rethink vector design. These are just some take-home messages. I won't list them all because I think it's just regurgitation. But one of the couple of things I'll just leave you with is that 30 to 70% of these patients have pre-existing antibodies for some of these vectors. So not only do we have problem in designing it, not only do we have problems making it, not only do we have problems finding these patients with rare diseases, but the large portion of these patients that we do find already have pre-existing antibodies for these vectors, which mean that they're not going to be eligible. So we have to find different designs again, or different capsids or different engineered versions that will bypass the pre-existing antibody issue. I think that's something I just want to mention that is outside of the scope, but it's very much a core problem in cell and gene therapy. And then finally, the price of cell and gene therapies are extortionate. I've touched on it there. The cost of goods, these must come down dramatically. If this sector is going to be successful for the next 10 years, we need the cost of goods to come down. And I don't mean a small bit, I mean a lot. So they need to come down a lot. And the only way I can feasibly think about doing that is through better vector design, as you see here. So what we're trying to do is rip up the playbook for conventional development and introduce a different Clinivac is what we're calling it, but it'll have different names for different people, of course. Extend the runway of your clinical design during the preclinical phases in order to de-risk and save you much money at a later stage. Okay. And that's it. Yeah. Thank you for your attention. We can do the Q&A session. We have loads. If anything, what I've said today, I've ran through a lot of it, of course, but by all means, have a look on our website and have a look. Thanks to Juraj for allowing me the opportunity to tell a story that not just serves, Cambridge, doesn't just serve VectorBuilder, but we're trying to serve the community, because a lot of people are not aware of this. They're doing hard work in clinic in research groups. They're doing hard work in academia, but not many people. Again, I don't want to self gratify myself, but there's not many people that have been in clinical trials and have come back the other end to tell the stories. So it's important that we don't have all the answers, but we have parts of answers and parts of solutions that we think can help. So hopefully that message came across, and happy to answer any questions you may have.